there. Thank you so much, folks, for logging in tonight. This is Popping the Science Bubble with Berkeley Public Library, and this is our first Popping the Science Bubble of the year. So we're just going to take a moment as folks are logging in. But thank you so much for attending tonight and listening to our wonderful scientists. Right. Yeah, not too much is going on at Berkeley Public Library, but I encourage you to always look at our events calendar. Next Saturday, we have our Lunar New Year event happening. It's going to be pretty exciting. We have 2 different lion dancing troupes coming to the library. So it's pretty exciting. I believe there will be food, and it should be pretty neat to watch. All right. Okay. That's going to be Saturday. Yeah, this coming Saturday. I think it starts at, oh my gosh, it's going to start maybe like late morning, afternoon. Year of the rabbit or year of the cat, according to the Vietnamese lunar calendar. So should be fun. All right, I'm going to go ahead and introduce tonight's program. All right, once again, thank you so much for joining us tonight, and welcome to Popping the Science Bubble. The Popping the Science Bubble is a monthly seminar series that aims to share new research findings from grad students and postdocs at UC Berkeley with the general public and create constructive discussion about a variety of science topics. We have 2 speakers at each seminar who will talk about their current research or a topic that they find really interesting. This is a partnership that the library has had with Cal for a few years now. And we're just always delighted to host the speakers once a month, every 3rd Tuesday of the month at 5:30. Right now, the program is on Zoom. We will let you know if it becomes an in-person program. And the older talks are archived on YouTube. The organizers for this program are right now 4 graduate students at UC Berkeley. Madison Lamore and Oksana Andrik have hosted the program for the past few years. And also joining us tonight are 2 new organizers, Kaylin Zoa and Daniel Bretthauer. Kaylin is a first-year PhD student in neuroscience, and Daniel is a second-year PhD student in astronomy. So you will be seeing more of them as they have joined the Science Bubble team. So just as a reminder, tonight's talk will be recorded on YouTube. I also highly recommend you join the Science Bubble listserv to keep informed on the newest talks. I'm going to hand it over to the Popping the Science Bubble team to introduce tonight's speakers.
Thank you for the introduction, Kelsey. And before we continue with the talks, just a few reminders. We encourage you to ask questions during the course of the talks using the Zoom Q&A button and the Zoom chat. And like Kelsey said, if you're interested in learning more about Popping the Science Bubble or checking out past seminars, you can visit our website and social media. You can see them here at the top of the screen on the right, or you can join our mailing list using the link or the QR code. We typically send 2 emails per month, one announcing the seminar and a follow-up. Our first speaker tonight is Jason, who is currently a PhD candidate in the Electrical Engineering and Computer Science Department at Berkeley. He grew up in Taiwan and came to the US after completing high school. He got his bachelor's degree from the University of Wisconsin-Madison in electrical engineering. Outside of research, he enjoys traveling and sports. He plays softball in the Berkeley Softball League and just recently got a puppy. We are very excited for your talk. Take it away, Jason.
Okay, let me share my slides. Okay, yeah, so the topic of today's talk will be beyond our usual fridge magnets and how to use magnets as a new building block for future computing hardware. So I will start with briefing a little bit about our current status of computing hardwares and what are the essential pieces. So here you can see in this picture, this is basically a very regular desktop computer that you will see if you open up a desktop. Where here you have a motherboard, which is depicted in brown with a lot of components on it. And among these components, you have the central processing unit, CPU. This is where all the heavy computation that happens. And then the memory units is another piece of important hardware that you will need to complete computations. And the last is the storage units, where data is permanently stored. So this basically build up of a contemporary computer. So if you open up your phone, your laptop, even sophisticated computers as servers in data centers, you'll see a very similar architecture, more or less the same with these three key components. And of course, you will need your input and output units, such as keyboard and mouse and your monitor to interact with the computer. So inside these computing hardwares lies this device called a transistor. This transistor is basically a switch that controls how much current flows through this transistor device. So if you open up any computing device from CPU, memory, or any storage devices, you will see billions or tens of billions of these transistors inside these devices. So on the left side here, pictured is a Intel's processor developed in 2007. This is a 45 nanometer technology. It's actually a pretty dated one compared to the phones or computers that you have today. However, compared to 40, 50 years ago, this is actually a very, very powerful machine. On the right is a transmission electron microscopy image of a single transistor. You can see this, this scale bar, which depicts 20 nanometers. This transistor is no more than about 40 nanometers in length. So what it does is you have a gate, source, and drain. And by changing the voltage at the gate, you can pass electrons from the source to the drain or turn off the path between these two source and drains. And that's how you control whether you have current flowing in a transistor or not. So transistors are everywhere in all of our computing devices. So I'll start from the left where I talk about CPU, where the heavy computation happens. This is mostly manufactured by companies that you probably heard about, including TSMC, Intel, and Samsung, but designed by companies like Apple, NVIDIA, Qualcomm, or AMD. So this is basically the brain of the computer. And the second is the main memory, where you need to temporarily store data when you execute computation. So for example, when you open up Google Chrome, the computer allocates certain data in the memory, either store or access these data from the memory and then grab it back to the CPU to do computation. So without this main memory, none of the applications that, including the Zoom that we're using right now, will not be happening. However, the difference between the memory and the data storage unit is that main memory, when you cut off the power or you turn off your device, the data stored in the memory is basically lost. So that's why we need data storage units to store data permanently. And that's also where our operating system is installed. So among these main computing hardwares, the one that I boxed in red are all composed of billions or millions of transistors, including CPU, main memory, and the solid state drives. So magnet is nowhere to be found in any of these devices, except the hard drive. Except the hard drive, which I'll talk a little bit more about. So transistor is the fundamental building block of almost every piece of computing hardware that you use, you interact, or you rely on a daily basis. We look at the market size of transistor-dominated computing hardware. It actually is pretty significant. In 2021, this amounts up to around $555 billion in the total sales. Just a quick comparison, the entire petroleum markets around the world is just a little bit higher than the computing hardware at 556 billion. So this is actually a very, very large market size in semiconductors. So what about magnets? Has anyone any idea or have any, think about how magnets can be used in computing hardware? Or even any daily interactions that you have with magnets? Okay, if not, it's fine. Let me give you a couple examples. So when I worked with magnets before my PhD, I was the only magnets I had interaction was when I was little, playing with these bar magnets or magnets from the fridge. But actually, if you think about it, there's actually a lot of application that requires magnets. For example, the car you drive every day. There's a lot of permanent magnets inside the electric motor. Without those magnets, the electric motors in your car won't be able to run. So that's a pretty essential one. So when we talk about magnets in computing hardware, let's look back about I think someone said using video monitors. Yes. So video monitors definitely has some of the magnets inside. So in the history of magnets, actually magnets is one of the first technologies that are used in storing information. So for example, credit card magnetic strips behind it is developed late in the 1960s. And it has been dominating the usage up till the late 1990s when microchips, which is also a form of semiconductor, took over. And now to a more contemporary style using contactless RFID chips. So that's a very, basically the first demonstration of using magnets to store data. And then we have magnetic recording tapes, basically cassettes. It's also around 1960 and 1990. These are actually very similar technology with the strips of magnets, but then it's replaced by CDs and now our digital age of streaming information. And next is the floppy disk. So this actually did not spend as much time around our lives, and also because it couldn't really store too much of data, and the usage is pretty limited. But nevertheless, it's still in the realm of computing, able to store data. So it was basically replaced by our thumb drives, also a semiconductor transistor-based device in the 1990s. The only one that has been survived up to now is the hard disk drives. So about 10 years or 15 years ago, hard disk drive is in every personal device. So if you have an iPod video, I think it's around 2005, that is probably one of the last portable device that has a hard disk drive in it. After that, it went into using semiconductor-based memory devices. However, hard disk drive is still the mainstream technology especially for a large amount of data storage, such as data centers or desktop computers. But it's starting to face some pressure from the lowering cost of transistor-based storage. And the market size has about 37.8 billion. And as a comparison to transistor-based semiconductor devices, it's about 555. So it's not even a 10%. So wire magnets are not used in the semiconductor technology historically. So to answer this question, there's a couple perspectives to look at it. Magnets are usually associated with a very energy inefficient way of storing data. The amount of energy required to switch the magnetization in a magnet is pretty power hungry. And the second one, which is probably the most important one, is the difficult to control the magnetic orientation in a very locally fashion. So in semiconductors, every device, every transistors are connected with wires. So we can easily access single devices through choosing an electrical path. However, if we still switch a magnet with a magnetic field, it's very hard to confine the magnetic field to only influence a very small region. It is not able to scale down to a very small footprint. So in that case, I'm going to show you, I'll kind of take a step back and kind of talk about how magnets are being switched in the first place. So magnetic moments, basically, if you think of it as a compass needle, when you apply a magnetic field, this moments will tend to align to the direction of the magnetic field. This is the most natural way of switching a magnet. So just like how we use a compass and we want to find the North Pole, the needle will align to the Earth's magnetic field. So this is not a scalable way. And actually engineers have utilized this method to try to build memory devices to serve as a memory technology, but it didn't work too well. So you can imagine what happens is that they have a very small magnet here. You can see a sandwich between these purple lines. These are all wires. And as they apply currents, the magnetic field can be generated and switch these magnets. So you can imagine that these magnetic fields have to be very controllable in a finite area or volume. And this is very challenging. So that means we cannot make these magnets too tiny, or we cannot pack the magnets in a very close fashion. So this limits our density. And when the density is limited, that means how much bits we can store in a unit area is greatly reduced. So switching magnets with a magnetic field is a non-scalable solution, and it's very energy inefficient. So we need a brand new way to switch these tiny magnets. So in order to talk about this new way to switch magnets, I'll resort back to an electron. So we all know that an electron carries a finite charge. That's how we have electricity conducted through wires. However, electron also has another property that is very interesting, which is the spin. So the spin of an electron actually carries a very, very tiny amount of magnetic moments. However, most of the time, the spins are pointing randomly to any direction, which when you sum up all the electrons, you don't actually have a net magnetic moment of a pool of electrons. However, if you pass these electrons with random spin directions through a ferromagnet, you can polarize these electrons in a certain direction. So we have a net magnetic moment within this pool of electrons. And if we pass this pool of electron that's been polarized through another ferromagnet, we can actually switch these moments. So these moments in the ferromagnet would like to align to the spin of these polarized electrons. And this term is called the spin transfer torque. This is a purely current phenomenon, not a magnetic field phenomenon. So what does this spin transfer torque, STT, enable? What enables that now we don't have to rely on applying a magnetic field, but instead, if we just apply a current through a magnet, we can control the magnetization in a very local fashion. So if you see this image to the bottom right, this is actually provided by Intel. This is a photo of a bunch of magnetic devices. So these black regions are metals where currents can be conducted. So if you want to switch this magnet over here, all you have to do is apply a current through it. And you can manipulate the magnetic moments in these areas. So we no longer rely on the magnetic field. Instead, we can use this current, so the density of these magnets can be higher. So the stack of this MTJ is on the order of 2 to 3 nanometers. So you can see the spacing between these two cells is probably only about 20 to 30 nanometers. That's a pretty dense memory cell. So where can we actually see MRAM in our daily lives? So this magnetic memory devices actually has already been commercialized, mostly by Samsung in the 2019. And we are most likely using some of these devices already. It's mostly used in devices that requires internet, but not heavily computing devices like our phones or computer, but small gadgets that require connection to internet, such as IP cameras or any smart home appliances that you may have, control your lights through Wi-Fi, these kind of devices, or even smart TVs has these magnetic memory units. Or your microcontroller units, which is usually used in consumer electronics that does not require too much of computation, such as washing machines. Or more recent vehicles also has these magnetic memories. And the last is the edge AI, which is AI accelerated chips. So since about 4 or 5 years ago, most smartphones start to have another dedicated processor to do AI computations. And this trend is continuing up to this date and will continue to grow in the future. And this is where the magnetic memory can demonstrate its strength. So where are we heading? So with the growth of this AI application drives the higher demand of these magnetic memories. You can see here on the right, this is the energy forecast of the amount of computation that will be used. So they project that in 2030, about 21% of the electricity generated are all going to be used in pure computations, in data centers, in your mobile phones, in networks. That's actually a huge amount of energy consumption. So trying to lower down the energy consumption of computing devices is a very pressing issue. And in a larger scale, this ties to climate change. So magnets can potentially solve this problem, being a very energy efficient way of computing data. So what are we working on here at Berkeley? So spin transfer torque is great, but there's still a lot to be improved. So I've mentioned spin transfer torque is already in commercial production, where some of the products that you are probably already using it. So the next generation is this called Spinorbit torque. This provides a much larger material space and an even lower power to switch magnets. And the most attractive one is that it can provide faster switching speed. So we can replace even more memory devices in computing hardwares. So this is a brand new way of generating spin in electron. So a quick recap for spin transfer torque: an unpolarized currents pass through a ferromagnet. And because all of these ferromagnets has a specific direction of its spin, they can polarize these electrons into its directions and become a spin polarized currents. So this relies on the requirement of a ferromagnet. And on Earth, we don't really actually have too many ferromagnets out there other than iron, nickel, and cobalt that we're all familiar with. So the material design space is much more narrow. On the other hand, the spin-orbit torque is a brand new way of generating spin currents by using many different materials that has this effect called spin Hall effect that can generate spin polarized currents. So what we're working on here at Berkeley, EECS, we are focusing on finding a very efficient material that can generate the spin polarized currents as depicted here. And this channel material basically can enable a different way of switching these magnets. So a quick preview of what we've done in this area. A couple challenges still sits here with the spin-orbit torque, mostly because of the compatibility with the silicon. So eventually, we still want to integrate these materials with silicon, which is where the money is at, and also where the capability is at the highest. So we need to find the materials that are compatible with silicon, and also there seems to be a trade-off between resistivity and the efficiency of generating these spins, and this is not good. What we ultimately want is high efficiency and very low resistivity. Because when we have large resistivity, that means we need to consume a lot of power. So in our department, we were able to identify a new material called Psilocyte that already has silicon in it, which means that there's great compatibility with silicon. And also it actually can create a large SOT, which is a large efficiency. So you can see in this plot here, one of it is iron silicide. It's above 1, which is pretty large, and at a much lower resistivity compared to these orange dots. And we're even be able to further scale down the resistivity while holding on to the efficiency, which is with cobalt silicide. So in our department, we're basically looking for new materials that can generate spin currents, and these spin currents can ultimately switch magnets in a very local fashion. So if you're interested, we have a preprint here listed with the DOI. You can look up, it's actually open access online. And with that, I'll conclude my talk over here. I'd like to acknowledge my group, my professor, Professor Salahuddin, my collaborators, and my awesome undergraduate researchers in our group. Thank you.
Great, thank you, Jason, for a very cool talk. It looks like we have a question. Do magnetic memory devices have to be isolated from other sources of magnetism, such as display monitors?
That's a very good question. So it's actually pretty surprising that these magnets are actually pretty hard to switch when we design these magnets, because you can imagine we have probably billions of these magnetic memory cells within just 1 or 2 centimeters footprint of a chip. So we can even worry about how the different memory cells within, the different memory devices interfere with each other. But we engineer it in a way that it's minimized by of the interaction among the different devices. So from that point of view, from a display monitors, you will have a little bit of magnetic field, but it won't be that much of an issue.
Thank you. As we wait for more questions, I was also curious. So this work sounds very interesting to me, and I was curious if you could tell us a bit about kind of what a typical day at work looks like for you, like what kind of techniques you use or like tools.
Yeah, so I should have included some of the pictures that I use for the tools I use. So of course, we're working with magnetic materials. So we have a lot of electromagnets in our lab. that can generate up a pretty big magnetic fields. So a typical day of my research in Berkeley is actually quite it's composed of a variety of different tasks. We deposit our own magnets in vacuum chambers, and we fabricate devices at the Berkeley Nano Lab on these chips, and then we measure them in these electromagnets through applying different currents. So yeah, for a typical day, it's basically spending in bunny suits in a clean room, fabricating devices, and operating these $1 million tools, and hopefully don't break them. And hopefully the device that we measure has behaviors that we expect to see.
If I can ask a follow-up to that, Jason, how much does a magnet's behavior change from that sterile lab environment to the kind of like actual device that it would be implemented into?
That's a very good question. So the magnets that we use in our lab are, since we're in the academic fields, we tend to not worry too much about how robust the magnets can last for a long time, but rather we focus more about the phenomenon, understanding the phenomenon of the material that we're studying. But of course, those magnets that are used in our phones for memory, those have to be gone through very rigorous developments. So the spin transfer torque that I just mentioned that got into commercial production by Samsung in 2019, it actually started developed in 2000. So it went through 20 years of research and development through many different companies, IBM, Samsung, all these semiconductor foundries, through rounds and rounds of engineering and improvements over time. So yeah, so it is definitely very, very different, much more robust when it has to come to, for a commercial product.
Great, thank you. Okay. If there are no further questions, I think coming through the chat and Q&A right now, we'll move on to the next talk. But Jason, if you'll stick around, if anyone has any questions that they think up, you can answer them in the chat, or we'll leave a few minutes at the end of the broadcast as well for any last minute questions to be asked. Oh, just kidding. There is one last minute coming through. Okay. An attendee is asking, does orientation of magnetic fields play a role in predicting their effects? If so, how do you do this? I don't know if do means kind of measure it or, study whether the orientation is.
Yeah, that's a very awesome question. So in fact, in one of my measurements, we actually do rotate our magnetic fields in 360 degrees, just because the angle between the magnetic moments and the current that is running through the moments actually spin out a angle dependence. So you're definitely right that the orientation of the magnetic fields and the effects definitely play a very important role. And especially when we're dealing with materials that are even more interesting, that has specific crystal orientations, there's another layer of complexity added on to it.
Great, thank you. All right. If I have not pressured anyone else to get any questions in under the wire, or you have a follow-up to that one as well, I think Jason will be sticking around. So thank you so much, Jason. That was a super cool talk.
Thank you.
Yeah, we're excited to have you have kicked off our 2023, and also excited to now have Elle Barnes here with us to talk about some salamanders and microbes. Elle grew up on Long Island, just outside of New York City, and has always been fascinated by wildlife and wild spaces, and Also, even more importantly, perhaps, how humans interact and affect those spaces, specifically studying how they interact with the microbiome, which are the microscopic organisms that live in, on, and around us. She got her BS from NYU and her PhD from Fordham University, both in New York City, and then hopped over here to the other coast to join the Joint Genome Institute at the Lawrence Berkeley National Lab for her postdoc. This department was originally formed to sequence the human genome and has since moved on into now filling in the microbial tree of life. In her spare time, she enjoys hiking with her dog, Gamma Ray, collecting plants and reading, and we're super excited to hear her talk tonight. So please take it away, Elle.
Thank you. All right, I'll go ahead and share. Okay. That out of the way. All right. So hello everyone. My name is Elle Barnes, and I'm currently at the Joint Genome Institute, still working on microbes, but actually in biofuel crops. If you have any questions about that, I'm happy to answer those. But today I'm going to tell you a story about salamanders, specifically the salamanders in New York City and their microbial friends, which was the work that I did as part of my dissertation. So the first question I'm going to send to the audience is, what is a mass extinction? Feel free to reply in the chat or in the Q&A. And even if you might not know the definition of a mass extinction, maybe you could suggest what perhaps is one of the most famous mass extinctions. Yes, I see someone said the dinosaurs. That's definitely probably the most common one that I see people say. So just to familiarize us, scientists, kind of estimate that throughout the history of the planet, there's been 5 major mass extinctions, and usually they're caused by some kind of cataclysmic natural disaster that happens, whether that's some kind of polar ice age, rising sea levels, volcanic eruptions that, cloud the whole atmosphere, or in the case of the dinosaurs, an asteroid that struck about 65 million years ago. And the real defining factor of a mass extinction is the extinction rate is about 1,000 to 10,000 times the background rate. And the background rate is usually something like 1 to 5 species a year, because of course, naturally, some species are going to go extinct and new species are going to form. So a mass extinction is 1,000 to 10,000 times that. But actually, that's not the whole story. And that scientists estimate right now, we're in the process of the 6th mass extinction. The difference here is that it's not really being caused by natural disasters, but are instead being caused by us, the humans. So the 6th mass extinction is also known as the Anthropocene, and here are just some of the major causes: overhunting, habitat destruction, pollution, climate change, disease spread. I'm sure many of you are familiar with all of these things, and some of those species that have gone the way of the extinction are, those such as the woolly mammoth, the dodo, and the passenger pigeon. But what a lot of people don't realize is that amphibians represent a group that is, especially of animals, one of the most vulnerable groups of organisms in this 6th extinction. And the IUCN, which is responsible for listing species for conservation concern, estimates that about 1 in 3 amphibian species is currently threatened with extinction. And the 2 major drivers are habitat destruction, where forests such as the Amazon are being converted either to agriculture or to an urban landscape, and then of course also disease spread. And I'll talk about these both in a little bit. But first, let's step back and just really understand what the world of amphibians looks like. So an amphibian kind of falls into one of these 3 categories. It could be a frog, a salamander, or a caecilian. The last is probably the lesser known. Caecilians are kind of like slimy snakes. So they are legless amphibians. And really in amphibians, there's over 7,800 species, and they've evolved over 400 million years ago. So they were existing before and alongside the dinosaurs. And they play a number of important roles in our environment. They link aquatic and terrestrial ecosystems. They play a role in food web stability. They help to indicate environmental change. And despite how diverse they are, how important they are in our environment, they receive 75 to 90% less funding than any listed mammals, birds, reptile, or fish from the IUCN. And one of the reasons for this is probably because they're a little bit elusive, and also people don't really understand a lot about them. But they need our help. They need some funding. And so I'm hoping I'll convince you today to maybe play a small role in the conservation of amphibians. And one of the big driving factors is disease spread. And this disease is caused by a type of fungi called the chytrid fungus. Scientifically, we call it Batrachochytrium dendrobatitis, or BD. There's also another species that's now called B. sal that is related. Essentially, the thought is that this fungi co-evolved on amphibian skin somewhere in Asia. But with globalization and the boom of the wildlife pet trade, which, lots of people want amphibians for pets, these amphibians and their fungi have hitchhiked around the globe and been spread to nearly every continent except for Antarctica, right? And in doing so, this fungus is no longer colonizing amphibians that have co-evolved with it, but instead is acting like an invasive species. And there, it's able to take over on their skin and cause disease. And one of the biggest symptoms, if you're going through the forest and you come upon an amphibian that potentially has this disease, is you'll see these brown splotches or lesions on their skin. This is a sign of chytrid infection. This is a microscopic image of a skin slice, basically the epidermis. These brown splotches are the actual chytrid fungi that are reproducing within the epidermis of an amphibian skin. Essentially, the fungus likes to eat skin. So it encourages the amphibian's immune system to upregulate the production of keratin, which is the main component of their skin, so that it can feed the fungus. And so goes the runaway train. And really what happens is the amphibian skin becomes really, really thick. And this is a problem because actually there's an entire group of amphibians that are known as the lungless amphibians that actually have evolved to get rid of their lungs and instead breathe entirely through their skin. So this is potentially very dangerous for those groups of amphibians. Eventually, this disease causes the amphibians to go into cardiac arrest and they die. And oftentimes, if you come across a dead amphibian that has chytrid fungus, they'll be belly up in some kind of aquatic habitat or in the soil. But today I'm going to be talking about this very cute and adorable and very tiny but mighty amphibian known as Plethodon cinereus, which its common name is the eastern redback salamander. They're very common to the northeastern part of the US and are arguably one of the most abundant salamander species in this region. And one of the most abundant species actually in deciduous forests in the northeast of the US. And they play a really important role in regulating the invertebrates, those bugs that live on the forest floor and help to decompose the leaf litter. While these salamanders eat the bugs and prevent the decomposition of leaf litter and instead help forests to store more carbon. So a lot of the carbon sequestration that happens in deciduous forests is actually due to these very tiny salamanders that are probably no larger than your thumb. So this is a view of the Catskills, which is probably the most pristine site in which you can find the salamander simply by flipping a log on the forest floor. But these salamanders don't only live in these types of habitats. Very interestingly, they also live here. This is very different than the pristine habitat of the Catskills. I'm sure many of you probably have seen an image like this before. This is Central Park in New York City. And believe it or not, these tiny salamanders also exist in these forested islands within where, pretty much they're surrounded by impervious surface. But as long as they have access to a water source and some kind of forested landscape, they can be found there. And this makes them a really interesting species to study how our changes in the landscape as humans are affecting other types of species. The other very important reason why they're interesting to study is they seem to be non-susceptible to the disease. And you might be asking, well, if you want to learn about the disease and you want to do conservation, why are you studying a salamander that's not susceptible? And the answer is that maybe if we uncover why they're not susceptible, we can help to treat those amphibians that are susceptible by uncovering Plethodon cinereus' secrets. So what is their secret? Well, it's kind of this complex triangle. Of course, we have the amphibian host, and on their skin, they're supplying sugary resources to microbes. These microbes could be the pathogen, the chytrid fungus, of course, that causes disease, which is called chytridiomycosis, but they also could harbor bacteria that could be helpful to them. And we call these symbionts. These bacteria are symbionts and helpful to the host because they directly can compete with the pathogen for space and resources, and thus indirectly protect the host from disease. But of course, there's this other side of the picture, which is the environment. And the environment is affecting not only how the host is responding, how their immune system might work and function, but also controlling which microbes are available to even colonize an amphibian skin. And therein lies the question: what happens if us as humans move on into New York City, completely change the environment, completely change the microbes, the bacteria, the fungi that might exist in those environments and be available to amphibians? And then how does that affect this thing in the middle? How does that affect competition with the pathogen? And then how are they protected or not protected? And that's kind of the question that I'm interested in asking. So the best way that I can model and describe this is, imagine you have some mainland, which is this blob on the bottom, and a new island arose, completely devoid of species. And you have a bunch of species, which are these colored shapes on the mainland. Well, when a new island pops up, they might want to disperse. And of course, inherently, not every species makes it to the island because they just don't have the traits that help to get them there. Maybe they can't swim or they can't fly. So already 2 species are knocked out. On the island, these species are going to reproduce, and some are going to reproduce better than others due to certain traits they have, or simply by random chance. And then, of course, they're going to interact with one another. We call these interspecific interactions. They're going to compete, or they're going to facilitate. Maybe they help each other grow better at the expense of another. And all of these things help to determine what kind of diversity you end up with. But what I just described is actually exactly what happens every day when a salamander is walking along the forest floor and coming up in contact with the soil. The microbes in the soil come in contact with the salamander's skin. Some can survive in a skin environment and colonize, others can't. And then once they're on the skin, they might interact with one another and do different things and change their population numbers. And this makes amphibians really interesting because they're islands of microbial diversity. You are also an island of microbial diversity, so you could just switch the salamander out, and the same would be true of your skin or your gut. So to cut to the chase, right, what happens if you sample the same salamander species in a rural habitat, a suburban habitat, and an urban habitat? And what you see is actually their microbiome, the bacteria, fungi that live on their skin, are very different. What we see here is a bunch of dots in space. Each one of those dots is a summary of their entire microbiome, all the diversity that's encompassed in it, composed into just a tiny dot. And the closer the dots are together, the more similar those 2 microbiomes are. What we see is that they completely separate in space, whether you're an urban salamander or an exurban, which you could also think of as a rural salamander. And the reason is that not only does their personal microbiome change, but the microbiome in the soil, in their habitat, has changed. And this could be due to things like how much canopy cover is there, how many trees are there, how much mercury is there, how moist is the soil, how much nitrogen and phosphorus might be there. And those little changes that can be significantly changed by human development, by urbanization, really do affect which microbes are available to colonize their skin and then actually do. Interestingly, what we found was that actually urban salamanders seem to have less chytrid, less of the pathogen than rural salamanders. This could be due to many reasons, one of which could be that urban environments are just really stressful. So maybe their immune system is just on high alert. But actually what we found is there's a complex thing happening here with their microbiome. So what I'm showing you here are 3 trees. And you might be saying to me, these really don't look like trees. Well, these are the types of trees that biologists like to look at. We call them phylogenetic trees, but basically they're family trees, where these horizontal lines are the branches. And the closer that 2 lines are together, the more related 2 things are. And we can build these trees with the DNA that we extract from the salamander skin or from the soil. And here we're looking at trees of bacteria. Now, the cooler the color means that bacteria does not fight the fungus very well. The warmer, the better it does. And what we see is that the color of the branches, depending on if you're in urban, suburban, or exurban, gets rearranged. And 2 that should be basically the same species, say 2 lines that are right next to each other on the tree, also could differ in their color. What this tells us is actually that humans can not only change which microbes are available, but in that habitat, we're also changing how those microbes might function. And this could get much more complicated, of course, but this is important if we're thinking about how can we help these amphibians? Well, it's not only important to know who is there, but how they're actually functioning on the skin. And that might not be obvious just from their DNA. The last thing that I'll kind of just talk about is this fancy term that's called alternative states. Now, we talk about alternative states, especially with the microbiome, when we want to know how stable a microbiome is. And this is important when we're talking about a pathogen. So if you just look at the diagram on the left, each of the microbiomes are represented by these balls, and these balls kind of fall into these pockets. You can have a healthy state microbiome and a disease state microbiome. But to move a ball from a healthy state to a disease state, just like in physics, you need some kind of input. And usually this input can be thought of as pathogen invasion. What happens when a salamander becomes diseased? It might be the push that a ball needs to fall into another state. Well, how stable that state is depends on how deep the pocket is. And so what we see here is that it takes very little energy to move a microbiome from a healthy state to a disease state, but to repair that microbiome from a disease state to a healthy state will require a lot more work. And this is something that we're finding in trying to treat amphibians that have disease. And so understanding how we can go from a healthy state to a disease state and back again is important. And what we found in our research is that this is highly dependent on microbial interactions. So which microbes are actually on their skin and how do they interact with one another? And what we found is that it's not only important to think about, hey, this bacteria can fight off the fungus because they produce a very specific chemical that engages in chemical warfare and kills the fungus. But what happens if that bacteria is grown alongside another competitor bacteria, right? Not all bacteria are just there to help and fight the fungus altogether, that they're competing with one another also. So this is a classic example of the enemy of my enemy is my friend, that the fungus sometimes can get a free ride and actually their abundance can increase in the presence of 2 beneficial bacteria because those bacteria are too busy fighting with one another that they're ignoring the fungus. And this is actually the question we ask when we're developing probiotics, which I'm sure many of you probably have heard about or simply read about on your yogurt label, right? How can we insert or augment our microbiome by adding certain live cultures of bacteria or fungi to our system? And this is the question that we're asking now. So the very last thing that I will kind of highlight is that I, of course, am not the only person working on this. And there are many wonderful people around the world working on amphibian conservation. And perhaps the most important people are those that are really working in parts of the world that are either underserved or hotspots for amphibian biodiversity, especially in South America and parts of Africa, believe it or not. So I've kind of included 3 wonderful people here that I encourage you to look up their research. Fortunate works in South Africa and is working with indigenous populations to identify how they identify species so that he can create field guides in many different languages, right? Bridging this idea that our experience with wildlife is tied to that cultural experience of wildlife, and that we have a lot to learn from indigenous populations, which many scientists have long ignored. Of course, Pedro is located in Brazil and works in many of the rainforest areas. and has been created this kind of organization called Documenting Threatened Species. He's an avid scientist, but also a photographer. Him and his students go out into the field and they document and tell the stories of amphibians that might be the last of their kind. I encourage you to check out their website. And then of course, also Teresa, who works in Bolivia, also in South America, and is responsible for a number of captive breeding programs where we're trying to take amphibians that might have gone locally extinct and breed them in captivity for the hopes of potentially reintroducing them in the wild. You might have heard of this water frog. He was very famous. His name was Romeo, and he lived for 10 years in captivity waiting for his Juliet. And through, believe it or not, social media and crowd fundraising, they were able to do a number of explorative kind of adventures into the jungle to find the last, what they believe to be the very last and very small population of water frogs. And in fact, did find his Juliet, and now they're working to try to breed this population in captivity. So in addition to what I've said today, I hope you kind of check out some of these other amazing amphibian conservationists that are working around the world, because this is a global issue. And with that, I will just take any questions and thank all these wonderful people that have been part of this research.
Great, thank you so much, Elle. That's a super cool talk. Lots going on just within the body of one salamander, so that's super cool. We'll let the questions come in through the chat and the Q&A as the audience have them. Okay, so to start off, one of our audience members is asking, how large an impact does fungi have on amphibian reduction in numbers? And can bacteria cooperate to fight off fungal infection?
So the chytrid fungi are actually, chytrid fungi are a large group of fungi, and BD and B-Sal, which are kind of the two most famous, are the only chytrid fungi that we know of that are pathogenic chytrid fungi. And there are a number of chytrid fungi that really serve no pathogenic purpose. And they can be very devastating. Within just a few years of chytrid being introduced to an environment, it could cause local extinction of an entire population of amphibians. And we've known about chytrid fungi for well over 20 years. And in that time, many species have since gone extinct globally as well as locally. So it can happen really, really quickly. And can bacteria cooperate? They definitely can, and that's something that we've seen. And actually, those bacteria would make really good candidates for a probiotic, right? Where you would want to use two bacteria that, like, combined can have a greater impact than just any one alone, and that's something that we're definitely looking into. The other thing that I didn't mention here, but is something we've also looked into, is of course the chytrid fungi isn't the only fungus. There are thousands of fungi that are also beneficial. That also exist on their skin. And so in addition to bacteria, of course, you could have fungi that are working together to fight this other pathogenic fungus.
Sorry, great, thank you, Elle. Super cool. If I could also ask a question while they're coming in. You showed the bacterial trees of different salamanders, and you were saying some of those species are the same species of bacteria, but less helpful in different populations of salamanders, right? Do you guys have any inkling if that's because, like, the salamander has a different diet, so it has different resource makeup on their skin, or is it the environment of the salamander? Like, I don't know if air pollution would make microbes less helpful, but kind of, you know, where that environmental change is causing that difference?
Yeah, for sure that, you know, each salamander itself is undergoing its own daily stressors, and that can change what it's exuding from its skin, and also that can change with the species of amphibian you're looking at. Of course, there are certain species of amphibians that can exude toxins, and that can totally change what the microbiome might look like. The environment definitely plays an important role. And I think the thing that's really cool about microbes, but very confusing to people, is that they operate on a different time frame and spatial scale than animals, right? So that salamander could, be moving around, its local forest, and from one forest to another, from one New York City park to another, you could have isolated populations of microbes that go through thousands of generation times in the lifespan or just a single year of a salamander's life. So evolution at the microbial scale is just so much faster. The other cool things about microbes is that they can engage in what's called horizontal gene transfer, right? So not only do they inherit genes from the mother population, but they also can cut out portions of their genes and pass them to their neighbor bacteria next to them. So they can acquire genes within the same generation from other microbial species or taxa. And because of these local things and the speed up of the time scale, we can get these kind of spatial pockets of very unique microbes that kind of genetically fall into the similar species group, but actually probably are, if you look at their whole genome, very different from one another.
Okay, so it's fully just different evolutionary species. At what point, I guess, would you consider the salamanders to have a different species of bacteria versus the same bacteria but different function?
Yeah, I mean, you'd have to, this is kind of the point of what we do at the Joint Genome Institute is because we're part of the DOE, we have a large swath of money to sequence everything we can find. Essentially, we can take that, scoop of dirt and extract all the DNA from it, sequence all of that DNA, and then using computers recapitulate the genomes, right? Individual genomes from that. And so from a single soil sample, we could get hundreds of mostly full, genomes. And this is where we're really starting to discover how little we know about the tree of life and how much more there is to fill in. Probably why, if I'm talking to a general audience, I'll say species, but if I'm talking to a very specific microbial audience, we don't really use the term species for microbes because we're not really sure it's a useful term anymore.
Well, thank you. That's super cool. And definitely a lot of data to crunch and a lot of existential questions about the tree of life.
Using lots of large computers, which Jason kind of alluded to in his talk.
Great. Well, thank you so much for your talk. If any last minute questions want to come through on this one as well, I think we'll have a couple minutes before closing. But otherwise, thank you so much for joining us for our first seminar of 2023. It's been a really great night to kick it off. And if any of our audience members are joining their first talk or still haven't yet, you can either check out our YouTube and or join our listserv to hear about future talks, as well as resources that tonight's speakers will have shared. In the chat, I've put the links to find both of those. So thank you very much, Elle and Jason, for coming out tonight. We very much enjoyed your talks.
Thank you for inviting us.
Yeah, thank you so much, Elle and Jason. Those were both really great talks. I did not know anything about these topics and I learned so much. I was just Googling stuff as you were talking and really interested. So really interesting talks. So thank you so much. Wishing you well, stay dry, stay warm, have a great rest of your week. And thank you so much, audience members too. Look forward to Popping the Science Bubble, third Tuesday of the month, 5:30 p.m. Please tune in. And these talks will be on YouTube. All right. Good night. Looks like we don't have any more questions. All right. Good night, everyone.